Hi, my name is Nadine. I'm responsible for business development for Ribbon Bio. Ribbon Bio is a synthetic biology company that specializes in DNA assembly, especially for long, complex molecules. Ribbon Bio wanted to address the challenges that come from synthetic biology. A lot of the time, chemical-based approaches for synthesizing DNA are more efficient and cost-effective and accurate for short sequences. But when the sequences stretch into longer stretches of DNA, it tends to drop off in terms of accuracy, and many companies have struggled to be able to synthesize DNA. Ribbon Bio has found a novel approach in assembling DNA that isn't typical for DNA synthesis. So we class ourselves more as a DNA assembly company, but this allows us to be able to work with those more complex, more challenging DNA sequences. Our flagship product is called MirrorSynth. MirrorSynth uses algorithm-based approach for DNA partitioning and then also enzymatic approach for DNA assembly. This, along with smart automation, has allowed us to be able to build a high throughput system, addresses problems such as being able to generate sequences that have high complexity, high GC content, and also regions that typically wouldn't be easy to synthesize. We also offer MirrorSynth in a cell-free version that also addresses issues that can arise from synthesizing DNA in a cell-based platform, such as high endotoxin. Ribbon Bio has been very active in driving collaborations and partnerships in the synthetic biology space, such as working with companies that can amplify the cell-free DNA that we produce in other cell-free platforms. This allows us to be able to work with companies such as Singoi that can take our DNA that we produce in a cell-free way and use their cell-free based platform to amplify the DNA for many different applications. Furthermore, we've also had collaborations with companies such as Scala. Scala focuses on developing high-quality proteins using our DNA. So synthetic biology right now is really exciting because of the diverse applications that we can meet and the needs that we can meet where previously we could not, such as cell-free approaches now are allowing us to develop DNA in a way that doesn't rely on cells. This way can be more economic, faster, and can also give therapeutic advantages, such as having less contaminants in the final product. This is something that we're seeing more and more in terms of the approaches that people are using in cell and gene therapy, but also in other sectors that you may not think are conventional, such as agrochemical. So right now, we believe that the industry is growing, such as it has in the last 20 years, but it's also diversifying. If you'd like to speak to us, we're always open to discuss both conventional approaches to DNA, but also some maybe more novel niche concepts. So please visit our website, contact us via the Contact Us page, and we'll be in touch.